Welcome back. And this is about the secondary structure of proteins. And there are two kinds of secondary structure identified for proteins: alpha helix and beta pleated or beta sheet. And here the alpha helix is the most common spiral structure of proteins, and it has a rigid arrangement of polypeptide chain. It was proposed by Linus Pauling and Corey, and for this Nobel Prize in 1954 was given to Pauling. And you can see when we speak about the structural features, it is a tightly packed coil structure with amino acid side chains extending outward from the central axis. So if this is a central axis running in a helical manner, you can see that this is the amino acid. Okay, like this is one amino acid, right, of which the carboxyl group has formed a amide bond or peptide bond with the nitrogen on the amino group of the next one. So likewise it goes. This is central axis, right? And the central axis, you can see it, from the central axis extends out the side chain, all the side chains. Okay, and this secondary structure of alpha helix do have, apart from the peptide bonds, they have the hydrogen bonds. These are the hydrogen bonds represented. Okay, and hydrogen bonds are formed between the hydrogen on the amino group of one amino acid with the oxygen on the carbonyl group or the carboxyl group of another amino acid, right? And each of this turn of the alpha helix contain 3.6 amino acids, and you can see 3.6 residues. Okay, 3.6 amino acids, and the width actually it is 0.54 nanometer. That's each turn. It may be adjacent turns may be 0.54 nanometer distance. Okay, and the spacing between two adjacent amino acids, it is 0.15 nanometer. So that is a alpha helical structural features. Alpha helix, it is a stable conformation and spontaneously formed with the lowest energy feasible, and we can have both kind of helices actually, left-handed as well as right-handed, but right-handed alpha helix is more stable than the left-handed helix. And we can see that when alpha helix is formed from a polypeptide chain, certain amino acids, the presence of certain amino acids can disrupt the helical formation. Okay, there are large number of acidic amino acids and large number of basic amino acids, and the presence of proline can actually disrupt the smooth formation of alpha helix, right? What happens when there is proline? Proline, we already saw that it is an amino group, isn't it? And whenever there is a proline in the amino acid chain, at that particular point there will be a bend, right? So that will actually disrupt the formation of alpha helix, right? Now there is more number of acidic amino acids. What happens when there is more number of acidic amino acids? Acidic amino acids in the sense, yeah, the polarity is changed, isn't it? I'm not speaking of the polarity, I'm sorry. So the charge, it is different, acidic and basic. So when there is more amount of specific charge, positive or negative, it is going to interfere the alpha helical structure. Okay, again, certain other kinds of bonds can form apart from the hydrogen bond, and this can disrupt the formation of alpha helix. Okay, so this is about the alpha helix structure, and I hope you remember that this is the main helix, central axis of the helix here. Okay, and the helix is formed of you can see here, it is the alpha carbon, the carboxyl carbon, and the amino nitrogen. Okay, the R group and the hydrogen, it will extend outward. Okay, hydrogen inward, and side chains will extend outward. Fine. And the axis is formed of N alpha carbon C, N alpha carbon C. Likewise, it will go. Okay, and the adjacent amino acids, it is placed with a 0.15 nanometer distance, right? And each turn of the helix, it contains 3.6 residues, and adjacent turns are placed 0.54 nanometer distant. And you can see that apart from the peptide bond in the secondary structure, apart from the peptide bond, they will have another bond, that is a hydrogen bond. And this hydrogen bond is formed between hydrogen and oxygen. hydrogen of one amino group or one peptide bond and oxygen of another peptide bond. Okay, so this is how the hydrogen bond is formed. And this hydrogen bond, when it is found, single hydrogen bond may be very weak, but when it comes to group, okay, aggregation, you can see here there are like more than one. And such a grouping or such a what you call extensive hydrogen bonding between the molecules, it can actually increase the stability. Okay, it helps in increasing the stability of the molecule. Okay, so this is about the alpha helix. When we speak about the beta sheeted condition, this is the second type. Alpha helix was the first one to be identified, and hence the second one which was identified was given the name beta. And it was proposed by Pauling and Corey. And beta pleated sheets, or some otherwise it is referred as beta sheets, they are composed of two or more segments of fully extended peptide chain. You can see the peptide chain is fully extended. Okay, and in the beta sheets, the hydrogen bonds are formed between neighboring segments of polypeptide chains, and it could be like the polypeptide chains in the beta sheets may be arranged either in parallel or anti-parallel. Okay. So here what happens? You can see side chains extend outward. Okay, this is somewhat like folded paper, isn't it? Somewhat like that. Okay, and how the hydrogen bond is formed? Hydrogen bond is formed between the, you can see here, between the chains. Okay, here it is anti-parallel. Why is it so? Because if this is the N terminal end and this is the C terminal end, you can see it is N over here and C on this side. And what about the next one? N is this one. N terminal end, this end is this one, and C terminal here. So that is why it is anti-parallel. Okay. What about parallel? Parallel do have N terminal on the same side. You can see N terminal, end of this particular chain, N terminal of this one, and it's all parallel. Okay. So that is why. What happens to the hydrogen bond? Hydrogen bond, it is almost parallel in the case of anti-parallel sheets. Okay. Hydrogen bonding is formed between the neighboring segments of the polypeptide chain. So this is one polypeptide chain, this is the second one, and the hydrogen bonding is formed between the neighboring polypeptide chains. Okay, and you can see the hydrogen bonding is almost parallel to each other. And what about here? It is slightly shifted. Okay, and another one is like, you can see it is 7 angstrom distant. And what about here? It is 6.5. So there is a difference in between the parallel and the anti-parallel. Okay, so the polypeptide chains in the beta sheets may be arranged either in parallel or anti-parallel manner. Okay. Parallel beta sheets are found in flavodoxin, it's a protein, and the anti-parallel beta sheet conformation is found in fibroin. Beta pleated sheets may be formed either by separate polypeptide chains or a single polypeptide chain folding back on itself. Okay, so if this is a continuous what you call a polypeptide chain, what happens? Okay, that this intra-chain what you call this one, the sheet. Okay, if there is interchain, that means several or separate polypeptide chains are there, and beta sheet is formed by the separate polypeptide chains. Okay, so this is interchain or intrachain, and it is stabilized by the hydrogen bonds between the chain. Okay. As already mentioned, many proteins contain beta pleated sheets, and alpha helix and beta sheet are commonly found in the same protein structure. So, for example, in the globular protein, beta sheet forms the core structure. Okay. So, this is about the secondary structure, and we saw that there are two kinds, alpha helix and beta sheet. Okay. Now, there are many other kinds of secondary structures, but most frequently found and the most stabilized ones are these two, the alpha and the beta. Now the next one is the tertiary structure. This is a three-dimensional arrangement of protein structure. And here you can see it is a compact structure with hydrophobic side chains held interior, while hydrophilic groups are on the surface of the protein. So when there is folding and what you call twisting and all, what happens is the protein structure or the conformation is maintained in such a way that the amino acids with hydrophobic side chains, okay, the hydrophobic side chains are extended into the interior of the protein, while the hydrophilic groups are extended on the surface. Because outside it is actually the protein molecules are actually interacting with the biological system which contains more of water. So hydrophilic groups are extended outward so that it can make polar interactions, hydrophilic interactions, hydrogen bonds, etc. While hydrophobic groups are extended inward. Okay, and this type of arrangement ensures stability of the molecule, and this stability is maintained with the help of the bonds, mainly the hydrogen bonds, disulphide bonds, electrostatic bonds, hydrophobic interactions, and van der Waals force. Okay, and this helps in maintaining the stability of the tertiary structure. Right. Now, regarding the quaternary structure, already mentioned, a great majority of proteins are composed of single polypeptide chains. But what happens? So there are certain proteins where there are two, which consist of two or more polypeptides, and these polypeptides may be identical or it may be non-identical, unrelated. Okay, these kinds of proteins which are formed of two or more polypeptides, they are known as oligomers. Okay, sorry, they are known as oligomers, and each monomeric unit which forms the oligomer, it is known as a monomer or a protomer. Okay, for example, a dimer consists of two polypeptides, right? So a dimer is a kind of oligomer which is formed of two polypeptides. If there are four, it is formed four polypeptides coming to form the oligomer, it is known as a tetramer. Okay, and tetramer will contain four monomers. I hope it is clear. When a protein is formed of two or more polypeptides, it will be presented, such kind of proteins are known as oligomers. Oligomers usually have a quaternary structure. Okay, and oligomer may be formed of same kind of polypeptides or different kinds of polypeptides. When an oligomer is formed of two polypeptides, we call such an oligomer as a dimer. And for example, it is found in creatine kinase. It's an enzyme, creatine kinase. It is a dimer, which is formed of two different kinds of polypeptides. Okay. Now, when it is formed of four polypeptides, we call it as a tetramer. Okay. So, likewise, it will go. Tetramer, we have lactose dehydrogenase enzyme. Okay, that is a tetramer. Clear? Okay. Now, what are the bonds that are found in quaternary structure that stabilizes the structure? The monomeric subunits are held together by non-covalent bonds, namely hydrogen bonds, hydrophobic interactions, and ionic bonds. So whenever we are speaking about all these bonds, we have to keep in mind that when you are asked to write the bonds found in a particular structure or particular level of protein conformation, you have to include peptide bonds everywhere. Okay, so basically there are peptide bonds everywhere, but apart from the peptide bonds, we are mentioning that is hydrogen bonds, hydrophobic interactions, and ionic bonds present in the quaternary structure. Okay, typical examples they are like hemoglobin, lactate dehydrogenase, etc. Okay, so when we speak about the bonds responsible for protein structure, there are plenty in number. The first one we can see is obviously the peptide bond. When we speak about the covalent bond, it's a peptide bond. Okay, peptide bond it is between the amino acids, so it forms a chain. It is found in the polypeptide backbone. Okay, so it is a peptide bond. And peptide peptide bond we have already mentioned. It is between the carboxyl group of one amino acid and amino group on the second one. So it forms a CONH bond, and the CONH bond, it is a covalent bond, which is considered to be the peptide bond. Okay. Now the next one we have is the disulphide bond. Okay, here you can see the disulphide bond. Okay, SS bond. Disulphide bond is yet another covalent bond. Okay, disulphide bond is formed by the sulfhydryl groups or the SH groups of two cysteine residues to produce cysteine. Okay, so one cysteine with having a cysteine molecule with SH residue and another cysteine molecule with SH residue comes together to form a disulfide bridge, the SS bond. Okay, and this particular molecule of two cysteines bounded by disulfide bridge, it is known as cysteine. Okay, so the disulfide bonds may be formed in a single polypeptide chain, or it may be between different polypeptides. Okay, these bonds contribute to the structural conformation and stability of the proteins. Right. So this particular structure, it is between amino acids of the same chain, or the same thing can be found in between two polypeptide chains. For example, as in the case of the immunoglobulin G, which you have studied in your immunology classes. Okay, so there it is disulfide bridges which bring together or stabilizes the structure. Okay, and it is between two different polypeptide chains. Now the non-covalent bonds. So we have seen that there are two covalent bonds. Okay, disulfide bonds as well as the peptide bond. Okay, so rest of the bonds we will be discussing in the coming video.